Back, the biology club is starting up, so some students are in charge of the biology club and looking for other students who are interested in it. And so you could scan that, or there are signs up in the hallway, I noticed, also in the science building. And this is what we're going to do, just updating this a little bit. So we're reading chapter 3, we're going through chapter 3, and and in chapter 3, 4, 5, the book has kind of moved over the years, moved some of the topics around, but really what we should be thinking about is we're doing a descriptive biology right now of the really early embryogenesis, and we're talking about the frog right now as a model amphibian, as an example of a vertebrate, and then we'll talk about chicken, and then we'll talk about mice/humans, mammals, and but we won't really talk about the embryo. What we'll talk about is some other parts of the mammalian development story, because the embryo at the phylotypic stage of a mammal like us looks just like the chicken we'll talk about. And so we're doing that, and that's descriptive, and then next week we're going to drop it down to the molecular level, and we'll just talk about the frog and talk about some of the proteins that are regulating those really early development steps. So as part of this, we're going to make a model of this chicken embryo, and I have a video, so I'll connect that video link with the lecture for today, and you don't need to scramble to write that down, because that'll be in the announcement as well. But I have some materials, art materials, to actually build a model chicken embryo, and the idea is it doesn't need to be fancy, you don't need to be an artist. The idea is this way to organize your learning. So it's all about learning. It's not really about building a model. It's about creating a learning experience that you can recall and over the years, literally. And that's because the early embryo of us and all vertebrates has these parts to it, like we heard about coelomic cavity. And those are just fundamental parts of our body. And there are features in the early embryo that lead to those adult features, and we should learn them as biology majors in the same way we know about legs and arms and so forth. So that's what that is. I actually was looking for that bag of stuff this morning, and I have a lot of different art materials, but that's my main bag, and I don't know why I misplaced it. So it's not out there quite yet, but it will be. Here's just an example of this coelom. So it's at our body cavity, where you can take any vertebrate, you can just slice them down the middle and flail open their body, and you can see all their organs, like their heart and stuff like that. Invertebrates don't have bodies like that. Lots of invertebrates do not have a coelomic cavity. If you think about real simple worms, like in the lab, if you're in the lab class, we're studying a flat worm called planaria. It does not have a coelomic cavity. On the other hand, some really simple invertebrate worms, like the earthworms that are crawling around outside, those do have a coelomic cavity. So the key thing for us here is just that it's our gut cavity, we have one, and all vertebrates have them, like that, okay? And so that's just a fundamental body part of all vertebrates. And so let's just roll on from where we're at, because at the end of class last time, we stopped talking about these early mesodermal derivatives, and they're like 5 fingers, and you can see this red or pink color. The notochord is surrounded by other mesodermal cells. So we're not talking about a notochord that could be isolated and dissected the way you could isolate and dissect, say, your intestine. It's not a separate piece of tissue, but it's cells that play a special role. And they can be subtly seen under a microscope, but it's not an obvious structure by itself, but it plays a role. And here's this cross section. It would be like a cross section through our body, and so that's that coelomic cavity that's starting to form, and down through the middle of it is our digestive tube, right? So our intestine goes right down through our gut cavity, and so that's that like sort of white U-shaped space is the coelom. Oh, that's not what I wanted to do. And why did it do that? That's weird. And so this would be the beginning of the coelomic cavity, and you can see that insects don't have it, if you want to compare it to one invertebrate. And then this would be the digestive tube, which developmental biologists often refer to the gut. And that's kind of confusing, because a lot of times we just think this is our gut, but the gut means the digestive tube. All right, so what we want to do, and we started doing this a little bit last Tuesday, is That's a cross section. If you turn, and here's a view of that turned. So if you turn from the side, it's like you're looking at your hands from your side, and you go from the rear end, the posterior, toward the anterior, the head end, and they're like it's like a finger, and that would be up here where the analogy would have like the fingernail. So it's, you know, not very clear from there. So what we kind of want to do is just turn this drawing a little bit and show that, for example, that notochord, if you turn it at an angle, is like a long hot dog. Ooh, that wasn't a very good line drawing. And so, you know, there would be a kind of shape like that if you saw it from an angle. And each of these 5 have that. And what we want to do is go back just a little bit earlier in development and look at those fingers. And they're initially just like 5 solid fingers. And there is a picture in your book that shows this earlier time in development. And so here are the 5. Well, it's showing a kind of a time in development that we want to focus on. So that's that notochord that's going to induce the central nervous system, and we haven't talked about that, but that's the central nervous system developing. And then here's one somite on one side and one somite on the other side. And here you can see the lateral plate mesoderm start to develop. And the lateral plate mesoderm are the 2 fingers, like that, solid initially, and then they begin to hollow out. And so they hollow out at their center. So you start with a solid thing like this, and then it starts to have a hollow center. And that hollow center is going to be the coelomic cavity. And we have one on each side, and they're going to merge together to make a single coelomic cavity. And so you can see that happening here. And so now we're going to go and look at this, and you can see that what's happening here is that hollow part in this lateral plate mesoderm has just gotten bigger, and the hollow place in the center of this lateral plate mesoderm on this side started getting bigger, and they've already merged together. So we see that, and they're just going to get more and more prominent. Now, in this picture, then, you can see that we could draw, and this is the lateral plate mesoderm, and I'll kind of color it in. So this line is the lateral plate mesoderm, and a coelomic cavity is actually partly defined by saying it's surrounded by mesoderm. So you can see the coelomic cavity is completely surrounded by mesoderm. See it? It's that stripe lining that, okay? So now I'm going to erase that and show you something. So you can see that this lateral plate mesoderm here is in a different position than this lateral plate mesoderm over here. And that those regions have 2 different names, and they have funny names, and I'll write them up. Maybe I'll draw it over here. So that inner part has a name that's surrounding the digestive tube, and then this outer part has a name. And the equivalent in our bodies today would be like when you do cut yourself and you flap your skin open, the inner part would be the inner surface. That's that part of the mesoderm here, and that region is pretty boring. It's just, you know, the inner lining of the thing, and it's called the somata pleura. So it's mesoderm, lateral plate mesoderm. This is all lateral plate mesoderm we're talking about, and it's just pretty boring. Then this lining, this has a funny name too. It's called the splanchnopleura. So again, these are initially mesoderm, then they become particularly lateral plate mesoderm, then become the splanchnopleuron and the somatopleura. And the splanchnopleuron mesoderm is going to be the mesoderm that gives rise to internal organs like our heart and kidney and some other internal organs. And we saw that the endoderm gives rise to some internal organs like the lungs, and the digestive tube organs like the appendix and so forth. Well, this mesoderm is going to make some really important internal organs. And so that's really special stuff. So the mesoderm is really great. Now we're going to just draw a picture like this just to look down, like from, like if you're lying on your tummy, and so you're an early embryo. So here you are lying on your tummy, and we can see that's the classic vertebrate architecture, right? So on that, you can say, in looking down, you can say, okay, well that's your coelomic cavity. It's in your body, your trunk. And so along the anterior posterior axis, we see this, that and this region, that lateral plate mesoderm, is forming the coelomic cavity. And then, just anterior to it, here, this is the region where the LPM in vertebrates that have limbs, fish don't have limbs, but other vertebrates do. In vertebrates that have limbs, the LPM later in our course we'll see plays a really important role in the development of each of those limbs. And you can see just beyond our coelomic cavity down here, we have that. So you can see the architecture of a vertebrate body is really constant. If it has limbs, it's going to be at the end of the trunk, and the limbs are basically going to be carrying around the trunk, the coelomic cavity, like that. Another thing to point out is that when we look, we see that when you look at a vertebrate, like just imagine a dog down on all fours, you can see here that the story of the vertebrate body plan has a lot of stuff in the dorsal region of the early embryo. So we have dorsal and ventral here, and it's really a dorsal story. We're going to have the notochord induce a dorsal central nervous system, brain and spinal cord, and then the somites are really arrayed here, and they're going to make the body bones. So we kind of have a rib cage that wraps down around. It's built on our spinal cord to protect our spine, but it also is built to just be holding the coelomic cavity wrapped around by ribs. And so really these mesoderms are also a dorsal body plan story. The dorsal mesoderm is really interesting and makes a bunch of really interesting things. And then the ventral mesoderm is pretty boring. It just makes kind of our tummy somatopleural, okay? So just looking at that and following up on that some. And then another thing I want to show you on this is that in the, for the side view, you can see that that there's this gray area. This is hollow, and this is the leftover part of the blastocele, and it's just going to disappear. And so remember that our first step in frog development was to make a blastula, and a blastula is a hollow ball of cells where the blastocele is in the center, and we're talking about a frog. Amphibians have a blastocele, but mammals don't, because our early development is different. And so that's showing you the blastocele, and you can see that disappearing. And then this would be that gray area there. That would be the coelomic cavity, or the coelom starting to develop. And this would be up near the mouth, and this would be near the anus, because this is a deuterostome, so we're setting up that body axis like that. And so that just gives you that side view to give you a little feeling of what's happening in the body then, okay? So, all right, great. And so here's just the words up here. You can, Google this and it will describe a little bit more. The splanchnoplura is just super significant in vertebrate body plans, okay? And so now, gastrulation. And so we've been talking about this process of gastrulation, and we'll say it's the beginning of morphogenesis because we start to fold the body together. The cells aren't really moving around very far in distance. They're just kind of sort of jostling. Like I said, if we brought our class to the middle, you wouldn't move very far, you'd just jostle, but that would change the shape of the class seats, like that. Okay, so we go from gastrulation to neurulation, and then we reach the phylotypic stage. So the next step is neurulation, and during that time the embryo is called a neurula, and all vertebrates do this. And what it's like is this, is that if we took that cross-section view, so this is that cross-section view where we can see the notochord and the somites and the lateral plate mesoderm forming, like that. Okay, so we're looking at that like a cross-section through your tummy. When we look at this, overlying the notochord, and I didn't draw that very well, but overlying the notochord, I'm just going to erase this and redraw it, just zoomed in on the notochord, because what we're going to see now is overlying the notochord is a layer of cells called ectoderm. And the ectoderm is fated to form the skin, the epidermis, and the central nervous system, the brain and spinal cord. And so now, underneath it, right, when we folded that mesodermal belt in, we folded it in, the finger of the notochord is now right here, and the notochord is going to signal these cells of the ectoderm to become the central nervous system. And so if we do a little timeline, what we see is those cells start to do this. They start to change their shape, and they make a shape that looks like this in cross-section. And then it starts to go like that in cross-section. And then it looks like this in cross-section. Sure, that was not very good artwork. Then it looks like this, where a tube, right? So this is in cross-section, so it's a tube like a garden hose. It has a hollow center, and that tube has pinched off. And the tube is long. It goes from anterior to posterior. And that's called the neural tube. And that's our central nervous system. That's our brain and spinal cord. And the brain and spinal cord today in our bodies is hollow. Our spinal cord inside the vertebrae, inside our backbone is the spinal cord, that part of the nervous system, and that's hollow today in our bodies as adults. That hollow center is filled with cerebral spinal fluid. So you might have heard there's a way of giving some pain medication at the hospital called a spinal tap, where they put a needle right into your back, between your vertebrae of your backbone, right into your spine, and into that center, hollow center, that is the cerebral spinal fluid, and they'll inject medication in there. Our brains are also like that too. It's less obvious, but here's a drawing of a brain in cross-section like that. And the center of our brains, there are some hollow regions, just kind of minor hollow regions that are called ventricles. And that's not your heart, that's your brain, and these are just called ventricles. And those are also filled with cerebral spinal fluid in any vertebrate brain. So those arise because we have a neural tube. And so we form the neural tube, and this is that stage called neurulation. And of course, it's super special for us because that's our brain and spinal cord, super special for any vertebrate. And that's what's happening during that neurula phase. That primarily is that, okay? And so you can see what's going on, right, is that, and we'll learn more about it, is that the signals that are coming from the notochord, they're received most strongly by these cells, and then a little bit less by another set of cells next to them, and less and less and less. And so we're making the neural tube by just being close to it. And you can see that there would be some cells, and we want to point out some really special cells that are here. And they actually don't get into the neural tube, so the neural tube pinches off, and these are cells that almost made it into the neural tube, but not quite. And so at this stage now, when the neural tube is formed, the ectoderm that's right above it, so all along our dorsal midline, is a set of cells. And those cells are only found in vertebrates, and they're in all vertebrates, and they're called the neural crest cells. And crest like the top of a hill, and you can see that's a great name for them because as these rise up, it's a little bit like the ridges along a valley. And the ridges are actually called the neural ridges or neural folds because they're going to fold over. And if you look down at the developing embryo, it would be right along their dorsal midline, and you'd see two stripes of cells folding up and folding over, and then the neural tube pinching up below. And the neural crest cells are cells that almost made it into the central nervous system, but not quite. And they're only found in vertebrates, and here's what the neural crest cells do. So if we look down at a vertebrate, and we say, here's its neural tube, and it's going to start forming the brain and the spinal cord. So that's the neural tube, and that's anterior to posterior. And then above it, so in this ectodermal cells above it, those are going to be skin cells. except right above them, right above the neural tube, there's now this kind of double stripe of neural crest cells. And in the evolution of vertebrates from the first vertebrate to the modern vertebrates, there's been tremendous evolutionary diversification of the neural crest cells. And they do several things, and we'll learn about them, but today what we'll just learn about is these neural crest cells that are on our back are going to crawl. They're not going to jostle, they're going to crawl, and that's called cell migration. They're going to migrate, and you see them coming up here like that and that up into the head region, and they're going to migrate and they're going to make the vertebrate face. And it's so dramatic what they do because, for example, that we have a skull, and you probably know that there are some like natural cracks in our skull. Those are called sutures, and there's one that kind of goes right like this, so we have sort of the front of our skull, and the entire front of our skull, and every vertebrate's skull, is formed by bone that is derived from the neural crest. And remember, we say that bone is a mesodermal derivative, but here's neural crest, an ectodermal derivative that's making bone. And remember, we showed the ultrasound of those dolphin embryos. Remember how different the shape of a vertebrate skull can be. Really big diversification of birds and fish and so forth in the vertebrate skull. And the neural crest plays a huge role in that. So it makes the bone, then it also makes a lot of the tissues of the face. There are some folks that are born with a birth defect that have a cleft lip or a cleft palate, and that's because literally these cells are migrating and they fail to fully zip together because the front of our face zips together. And we'll learn some about that in future classes, but for right now, the neural crest cells, they make more things we'll learn about. For right now, they build a vertebrate face. One thing about that is fascinating is that all vertebrates have faces. Fish and everything have a face made by the neural crest cells, and invertebrates don't. Invertebrates have a head, like a fruit fly, and insects, they have a head, but they don't have a face in the same way that all vertebrates have a face. And so that neural crest cells, those are probably the most significant evolutionary feature of vertebrates that make us distinctive in the roles in that role. So that's something about neurulation, and those neural crest cells are emerging here, and they're starting to make a move, and one of the things they're doing is they're heading up to make the vertebrate face. So the neurula is a really famous time, and then we're set. Then we reach the phylotypic stage before we have other body parts. No heart, no lungs, no limbs at phylotypic stage, okay? So that's great. And so let's just look at this list, because this is a list of examples that we want to learn about for the vertebrate phylotypic stage. And we'll learn more than this, but we just be like, do. And the order of the list doesn't matter, but these are some important features. And it's really the focus of the assignment is to build a model and label it with these and some more things, but these are the big ones. So notochord, we're set with that. Pharyngeal pouches, we haven't mentioned those, got to mention that. Deuterostomes, mouth form second, triploblasts, 3 germ layers, somites, those are going to form, they're going to slice up into numbered somites and form the vertebrae, so they're going to form bones and muscles in our body. Lateral plate mesoderm, the splanchno per splanoplural, the coelomic cavity. Then here's a special embryonic name we haven't mentioned before in our course, and it's just a special term, the term for an embryonic digestive tube. So I mentioned developmental biologists typically call the digestive tube the gut. The development of the gut is the digestive tube. There's this official traditional name, archenteron. And then we just talked about the neural tube and the neural crest cells. So we're in really good shape with the exception of the pharyngeal pouches. And they're also called branchial arches, and they're here. My niece, Emily, that had a sweaty neck, had an issue with the development of this. And in this region, our lower face, there's tremendous evolutionary diversification that has occurred in vertebrates. And so here's just a picture of that. So The original vertebrate, it's really hard to imagine this, the original vertebrates had a mouth, but it didn't have a jaw. It just was like, and these are these branchial arches, and you can see they immediately remind you of the gills of a fish. So that's one thing that the branchial arches evolved into. Other animals, other vertebrates don't have gills, but one thing they developed into is that. And then you can see they also developed into the jaw. So the branchial arches, the first branchial arches evolved into being a jaw. And that's a big part of being a vertebrate. And so this is just a little hint of the products of these branchial arches. And they're right in this area, and you can see there's some really famous things that form from the branchial arches. So like the little bones of our inner ear are derived from certain branchial arches. And we don't need to learn any of these details now or in our course because it's really complicated. A lot of stuff happens there, and it varies. There's some vertebrates that have inner ear bones, and some don't even have ears. You know, so there's a lot of evolution that's taking place here. Another thing is, if you've taken human anatomy and physiology, you probably learned about the cranial nerves. So there are 12 cranial nerves, and really famously, one of the cranial nerves is involved in each of the 6 of the branchial arches, and they do a lot. There's just a lot of evolution that has occurred in the development of those cranial nerves and how they pass through our skull and stuff like that. For us, for now, it's just that's what's happening, and that's a really big feature of early vertebrate development is these branchial pouches. So we want to get that on your model. The other reason we don't talk about it much in here is because there's actually a course called Comparative Vertebrate Anatomy that deals with the evolution of vertebrates and the representation of modern vertebrates and how there's a variety of modern vertebrates like fish and people, and then the history of that. And Rachel Simons, who's awesome, teaches that. Has anybody in here taken that course? I think it's taught every other spring. And I think it was taught last spring, so it won't be taught this spring. But Rachel's great, and it dovetails with this course. Rachel's a physiologist, so Rachel's looking at the product. We're developmental biologists, so we're looking at the creation of these. And so that's the branchial pouches. So let me just go down to this, back to this list. And with that, I think we're really kind of set up to do the assignment. There's more information that comes out in the assignment like that. And so we'll build that, and it's due a week from today, where you'll build it, and you don't need to bring it into class, just take pictures. Another thing I didn't mention is you're welcome to work on these assignments together. You each have to turn in pictures of your own model, but absolutely you could work with a group of people, and sometimes students do that, where I'll have these art supplies out, they'll go over there after class or sometime, they'll meet there, and there are some tables nearby, they can just build their models together. That's great too, because again, that provides a memory that you recall. Oh yeah, I remember the model, and I remember where I built it, and I remember I built it with, Nick and Jennifer, like that, right? It's just super helpful to do things like that, rather than, just going off to the same little corner of the apartment, and or your apartment or the library to study, okay? So your model's gonna look like something like that. That's this fancy example of it that worked out great for a student that had a lot of art skills, much more than me, okay? All right, so on we go, and we talked about that, and that's all about this phylotypic stage that would be representative of all vertebrates. The model you build will be what you look like at one point in your life, okay? And it goes in here, so we're talking about converging. We started in a different place. Now we're going to turn to the chicken, and we're going to start out with an even bigger egg. With the frog, we had a big egg, and the entire egg really turned into the embryo. With the chicken, the egg cell is that whole yellow in a chicken egg, and it doesn't all turn into embryo because there's so much yolk. Reptiles and birds, lay that hard shell, and everything has to happen inside there for a chicken to hatch that can get up and walk around and feed itself. So mom has to pack in extra nutrients, extra regulators in this chicken embryo like that. So returning to that, and what we'll see is that instead of developing as a sphere, it develops more like a coin, like a disc or a frisbee on the surface of that yolk, just a little tiny dot. And you've never seen that inside an egg you break at home. Sometimes you see some blood. That's from when the chicken ovulated the egg. And sometimes you see some little funniness in there, but it's not the embryo, because these eggs are not fertile eggs. They don't have a developing embryo. So let me just come back to that. But so here's just a picture like that of fertile egg and what it looks like when it's developing. And you really can't see it here, but there's just a faint little gray dot. Kind of like the dot of a pencil eraser would make on it, about that big. And there are some old-time descriptive terms in describing it. So the center is called the area opaca, and around the margins is called the area pellucida, and that's going to develop as an embryo on the surface. And so we're going to imagine that the embryo is just lying down on its tummy, with its head over here and its tail over there, like it's a coin on the surface of the egg yolk. And so this just shows the start of that development. And so we have these two areas, the central area pellucida and this area opaca is like that. And what you see is at on one end of this, you start to see a little groove forming. And it is physically a groove, and it's called the primitive streak. And that's going to be the equivalent of the blastopore. That's where cells are going to fold in during gastrulation. And so start to imagine like it's a little wrinkle in a blanket, or you could think of it as like a little valley that's forming. And so because the primitive streak forms and it appears like it kind of migrates out, you see smoothness, and then it starts to form a dent, and the dent progresses out, not quite halfway across it. So we're looking down, and we're seeing that early development. And so this is where right about the time the chicken lays the egg, that's what's happening inside the eggshell. And so on this, there's a feature, and that is at the sort of the tip of this, the front end of this, instead of being a valley, there's a bump, like a little hill. So now you're standing down here, and you're looking up along this valley, and up along it, there's a valley with, sort of hills on the side. And then at the far end of it, the valley is getting longer and longer and longer. And then beyond that, there's a bump. And the bump is called the node, and it's called the node in mammals. In a chicken, it's called Henson's node. And that forms, and then we get, so that's called forming the primitive streak. And then we get the next stage is the stage that's equivalent to gastrulation. So things are going to fold together, and it's called primitive streak. So there's the words primitive streak regression.
The node is the black dot in the center?
Yep.
Okay.
Yep. And so what primitive streak regression looks like is, it looks like that node, that hill, is going to come at us along the valley, and as it's going along the valley, it's covering up the valley. Another way to think about this is, if somebody's going to plant some seeds in a garden, they'll take their hand and they'll make a groove, they'll throw in the seeds, and then they'll take the dirt from the groove and they'll just cover up that. So that's what's happening in primitive streak regression, and that's gastrulation. And so in the process, imagine this. Imagine that the node is like a glacier coming down the valley. And it's just pushing it all down the valley. And as it's moving down the valley, we see along the valley, the trees on the hills on the sides of the valley, those are folding in. So cells are folding into the valley like that. And then the glacier's coming down and just covering up the valley. And behind the glacier, everything's smooth. As that glacier's coming to the valley, if we could see it, what's happening is that node, in the node, those cells are going to be mesoderm, and it's dividing really fast. So it's a small little hill, but the cells are dividing, and it's producing those. And on the front of the glacier, as it's coming down the valley, you can see like chunks of the glacier are just falling off. Those are the new cells. And they're getting covered up as they go forward. So we get two sources of cells that are getting covered up. The sides of the valley, of the primitive streak, are folding in. And then the front of the glacier is dividing really fast, and those cells are getting folded in. And so if we look at this drawing, what it's like is behind, and maybe I'll draw this a little bigger because you can see it better. So, you know, when we were talking about the frog, a lot of times we want to make cross-section. We make a cross-section to look at the fingers like little circular slices. Here we're looking down, so it's again like you're laying on the floor on your tummy, and we're going to build an embryo. So we're going to have anterior, posterior like this, and left and right like that. And so if we look at primitive streak regression, we have this area like this, and the primitive streak regression has that node, or Henson's node, and you can call it either one. We don't need to make that distinction here. So the node is coming down the valley, and things are getting covered up. So we have mesoderm and some mesoderm, some endoderm that's folding in like that. And then we have lots of mesoderm that's folding like that. Then behind the valley, or behind the node, that's here, and behind the node, we would draw these cells kind of like a dotted line because they're covered up, and they're underneath the ectoderm. But that would be the notochord.
Oh, okay.
And so this would be the anterior end of the notochord. That's going to induce the brain, and it's going to induce the cells that are above it, that's the ectoderm, to form the brain and then the spinal cord. And then we have next to those, we have those somites, and they're initially a hot dog finger. They get divided up into the somites, and they're numbered. They're just named by numbering 1, 2, 3, 4, 5, and so forth. There's 40 of them, something like that. Varies in different vertebrates, some. So those are the somites, left and right side, and they're going to form the bones and the muscles and other parts of the mesoderm. And then flanking them, we have the lateral plate mesoderm. And so that's that, and that's going to start to form a hollow along the trunk area to form the somic cavity. So that's what that looks like. And when that happens in a chicken and a mammal like us, what happens is just in a single embryo, we see a big range of development. So remember in the frog, we said, oh, first we're going to make a blastula, and then gastrula, and then a neurula. In one chicken embryo, we can see the earliest stages. It's not a blastula, but the earliest stages forming the embryo. And then we can see this, which is the equivalent of gastrulation, and then we can see this, which is the equivalent of neurulation. So that's one reason why we're making a chicken embryo model, is because from anterior posterior, you can see in just one model a big range of developmental stages. If we just redraw this and zoom in a little bit, what that would look like is, now we'll make the node big like this. So here's a big node. And so then behind the node, there would be this dotted line that's going to be the notochord, like that. And then if we drew the cells above it, well, these would be the neural crest cells. And then this would be the place where the neural folds were happening. And then this would be a place where they haven't even started folding yet. So along that length, we see that progression of the neurulation, where just anterior to the node, the ectoderm is just smooth. And then you see these dents or ridges forming and folding over to make that neural tube. And along the length of the chicken, you see the brain forms first, and then we just zip together a neural tube from anterior to posterior. Okay, and so this is an image from your book, And it's showing that coin shape, it's trying to show the primitive streak, like that, showing how those cells along the line fold in. They're going to be endoderm and some of the mesoderm. There are lots of terms here, and the key terms I would say is their coin has like two sides to it, and this upper side is the epiblast, and that's going to give rise to the chicken embryo or to the human. And then the lower part of the coin is called the hypoblast. That's going to give rise to what are called extraembryonic tissues. So inside a chicken egg, the little chicken is growing inside a bag. That's true in mammals too, right? There's the amniotic sac around the baby. If we just think about mammals for a minute, when a human gives birth, they give birth to a baby, and then in the next hour or so, they give birth to what's called the afterbirth. And the afterbirth is a bunch of tissues as big as the baby that represent the placenta and the amniotic sac and some other membranes. So we have, in mammals, we have embryo, os. Remember these extra embryonic tissues, the most famous one is the placenta. Those extra embryonic tissues are actually composed of some cells from your mom and some cells from you. So in mammals and birds, there are these extra embryonic tissues that the embryo contributes to. And so that becomes part of this story. And we're really interested in that upper epiblast surface because that's going to give rise to the embryo. So just more images from your book about that. And then this figure used to be in the book. It's not there anymore. But that's the figure we use for our model. And here what we have is there's the primitive streak back here in the posterior region. There's the node, or Hansen's node. It's regressing, going from anterior to posterior. Right anterior to the node, the ectodermal surface of the embryo is smooth. And then we see the neural ridges rising up. They've indicated little bumps where the somites are starting to appear. And then if you look at the cross section here, we have the neural ridges, and this is called foreplate, and the neural tube. There's the notochord inducing it. There are those somites. There's the lateral plate mesoderm starting to form this lumen cavity. We go more anterior, we can see further development and more advanced development of those stages. And so really no new terms. This is just a figure from another book that's trying to show that although we're starting with a flat coin like embryo in chickens, you could just pretend to take the coin and fold it, like sort of like a bowl that's upside down. And if you did that, the cross section of a chicken embryo and a frog embryo actually look pretty similar. A key difference is that the frog goes through that blastula, gastrula, nerula stage, so really distinct steps. Whereas in a chicken or mammal, chicken, reptile, bird, or mammal, those stages happen within the embryo at the same time, where in the most posterior region it's earlier in development, and the more anterior region it's later in development. And with that, we're really pretty good in covering the chicken. There are really no new parts to add except for a couple things like the epiblast and hypoblast, okay? Let me just go back here because I skipped a couple of simple slides to show you this. And I just wanted to show you this, just a little add-on, nothing to learn here, but just so you're aware of it. So we talked about in the frog embryo, and there was a couple of questions about that, about slicing the frog embryo, right? So here's the fertilized egg, the one-celled zygote, and it's going to start dividing. 2-cell embryo, 4-cell embryo. So it's doing that. And they divide in a distinctive way. So first a vertical slice, another vertical slice, then a horizontal slice. And this guy and other people 200 years ago looked at the first cell divisions of all fertilized egg of all animals. And they found that they fit in some different patterns. And then related species have the same kind of cell division pattern. And so this is from a different book, and you don't need to see the words, that's fine. But just see that that's the first single-celled embryo. You can divide it to 2 and 4. And you can see they're dividing with some different patterns. And the point of this slide is that all animals fit into certain patterns, and all the animals that are related fit into the same pattern. So it goes back to that indication of evolutionary relationship between similar species. And I'll just show you this. So for example, this one, the way these guys are dividing, they're dividing kind of on the surface like a coin, like a chicken. So there's chicken. Birds, fish, reptiles all have these giant egg cells, and they make an embryo on the surface, like that. Here's one, this is fruit flies, so a different one where they divide differently. We'll learn about that after our first exam. Here's an example of mollusk, here's amphibian, like the frog we talked about, and here are mammals. So mammals are very different. We don't have that big egg yolk, different type of cell division. Here's some annelids, here's some echinoderms. So that all animals that are related fit within no exception into that pattern of cell division. It's just another example of the diversity of animals, the diversity of animal embryos, but the similarity between embryonic development in closely related animals. All right, so we're just gonna roll on. Do people have questions about that? So the next thing to say is that human and early embryos up to that phylotypic stage look just like the chicken. It's the same deal. We look like a coin, okay? And I just show you a picture of that. This isn't from your book. This is just to give you an orientation. So this is mammalian fertilization reproduction. So there's the ovary, there's the fallopian tube, there's the uterus. And the female ovulates an egg, and it's moving down the fallopian tube, and it's in this region where it will get fertilized. And this will be, well, our example will be a human. For the egg, it takes about a week for it to be ovulated and move to the uterus. It varies some, and during that time, anywhere along that time, it could get fertilized. So the timing of fertilization varies a little bit, just a little bit. But we're talking about just like the first week of development in a human embryo. And so we'll get ovulation, then fertilization, and then this is called implantation. And implantation takes a few days after fertilization. It varies. Could be a couple days, could be a week. So we get fertilization here, and this is the zygote. So we get a zygote, the fallopian tube, and we start to get a 2-cell stage, 4-cell stage, 8-cell stage, and the embryo is just a solid ball of cells. So it's very different looking than an early chicken or frog embryo. It's tiny, there's no yolk, and it's just tiny little set of cells. Then it implants. And here it is implanted. It starts to develop. The embryo starts to contribute a lot. You start to contribute a lot to these extra embryonic tissues. And so we start to see this sort of nodule in the uterus developing. And if you look inside of it, and this is color coded, inside of it, there's a disc, and here it is the embryo developing in that disc. So the embryo is just a little tiny dot inside this larger tissue that's developing to form the placenta and the amniotic sac in the uterus. And this just shows there's the node, there's reproduction, so that line would be the notochord, somites, lateral plate, mesoderm, forming in a mammalian embryo. And the book goes into some detail in talking about the mammals, but the book goes into details that aren't helpful. So here's what we're going to learn about the early development of mammals. So the first thing is that there's a stage after fertilization that's called pre-implantation. All right? And we're going to talk about that a little bit. So the pre-implantation embryo is there for just a few days. It varies depending on the time of fertilization, but it's just there for a few days. And so in mammals, this pre-implantation embryo is going to be a solid ball of cells. That forms a little ball called the morula. And the cells, here's a 4-cell stage embryo. So during this implantation embryo, we're getting the cells dividing, and so we got, 1 cell, 2 cell, 4 cell, 8 cell, like that, okay? And these cells are all equivalent. So at any time, we could split the embryo anyway, and we'd get twins or triplets or quadruplets. And during this pre-implantation time is when identical twins form. During this time, the cells are equivalent. So if we split it, we can make 2 embryos. If we split it more, 4, more and more and more. Okay? This is the stage when all the cells in the embryo they're all called embryonic stem cells. And embryonic stem cells only occur in mammals. And they're fascinating because each cell has the potential to make the whole organism. Because of that, we can say that when these cells are dividing during this pre-implantation period, they're dividing like this to make more cells. This is an embryonic stem cell. Remember how we drew this out for the adult stem cells, like the satellite muscle stem cells. When these cells divide, they divide symmetrically. And so we're increasing the population of stem cells with these symmetric divisions. Remember, in our satellite muscle stem cells, they divide asymmetrically to make a self-renewing stem cell and then a precursor cell. We're not making any precursor cells, we're dividing symmetrically. So really fascinating. You can take this embryo out and put it in sterile culture in a dish, in a lab, and you can just get it to expand indefinitely. And that's what people are really interested in when they study embryonic stem cells. One thing just to mention is to see that the cells are only present during the first week. And so it's impossible to get embryonic stem cells from any person that has had an abortion. Those tissues don't have any embryonic stem cells. They have adult stem cells because they're later in development. And so it's just a good question to say, where do these cells ever come from? And the answer is there are families that go through in vitro fertilization. And so they're having a hard time getting pregnant. And so in vitro fertilization involves taking the egg out from mom and sperm from dad, joining them together, fertilization in a test tube, and then implanting those in mom's uterus. That procedure doesn't work very well, so anytime it's done, mom is given medicine to superovulate that month. So instead of releasing 1 egg, mom might release 8 or 9, and they're all collected, they're all fertilized. The early development is allowed to occur just a little bit in the lab, in the clinic, and then a couple of those will be implanted. Because it's inefficient, a couple will be implanted. As a result, lots of IVF gives rise to babies that are twins or triplets because they put 2 or 3 fertilized embryos in. Those are not identical twins. Those are fraternal twins. They're like siblings because the 9 eggs that got ovulated and got fertilized are all separate, and they're like 9 different babies. So let's say 9 are fertilized and 3 are put in. The other 6 are just frozen in case the procedure doesn't work. And that's the source of embryonic stem cells, because the mom and dad have as many kids as they want, and they're like, we're done. And you're like, what are you going to do with those embryos? And they're these very, very early embryos, 128 cell stage embryos like that, very, very early. And they might say, well, we don't need them. And they say, you want to donate them to research? And even if the 1 embryo is donated in the dish, the scientists can expand that number by that symmetric cell division to make a lot. So there's a lot to say about embryonic stem cells, and so our book dives into that a little bit, okay? And we just want to be careful because we're still wanting to do descriptive embryology, and we don't want to get too derailed by that. But we want to learn a little bit about embryonic stem cells today, and then we'll learn more about them late in the semester. What's the next thing we want to say about them? And that is that the book talks about twins, and it talks about I don't even have that figure in here. I should have it somewhere. Just don't have it. The book talks way too much about the extra embryonic tissues, and those become really complicated story. So here's what we're going to do, is we're not going to pay attention to the book in chapter 3 talking about all the different aspects of the placenta and the folding together of those tissues. And instead, we're going to focus on this figure and learn just these terms. So what this shows is it shows an early mammalian embryo, and it's going to develop further. So it's a little bit of a timeline. And so it's already past this morula stage, and it's just beginning to implant, and that is called the blastocyst. And so let's learn these three terms. That, yeah, you start off fertilized egg, embryonic cell division gives a little ball, a little morula, that develops further into a blastocyst, which is just a ball. And then we start to be able to distinguish between inner set of cells in the ball and an outer set of cells. And the inner set of cells is called the inner cell mass, the outer is called the trophoblast. And they're significant partly because they're the first cells that start to have a really different role in the development. So for example, if you look at the trophoblast, the outer cells, you don't need to learn those terms. Those are some extra embryonic tissues. So about the time of implantation, the embryo starts to start to have some differences. That means they are no longer embryonic stem cells. These were dividing equally and could make any part of the embryo. Now the outer cells can only make some of the extra embryonic tissues. So that's where we have the ending of embryonic stem cells. Then the inner cell mass, it's going to make the entire embryo, but also it's going to make a lot of extra embryonic tissues. So that's really important, that right about the time of implantation, we start having embryonic stem cells. They start to have limitations of the types of cells they can produce. Yes, they are still have genomic equivalents, but they just don't have that same ability to make everything. So we want to learn a little bit about that fact, and just learning these three terms, right? Because of that timing right around implantation, when the embryo shifts from being able to have every cell make everything to start having some restrictions. And that's why we want to focus on that. So we'll learn those three terms, and we won't get strangled and derailed by all the details in the book, okay? Then there's one more piece. So kind of the three pieces we want to get out of mammalian development, or you could say four. First, the embryo looks just like a chicken at the phylotypic stage, just like the model you're building. Second, right after fertilization, the cells are all equivalent in mammals, and they're embryonic stem cells. Each cell could make the whole organism, as evidenced by identical twins. Then third, there's a time right around implantation where we start to see differences between what the cells can make. And then fourth is one more thing we're going to talk about that our book talks about here, and it's an experiment like that. Instead of really just describing our natural development and natural mammalian development, it's an experiment that scientists find really, really valuable. And our book talks about there, so we'll cover it there too, because we have the knowledge to be able to understand what they're talking about. Let's do a little concept chat action here. And so try to find a partner. If there's three, look for, splitting that group up. I have not handed this out, so Zach is going to get really good at that, grabbing that. And so make sure you sign up. That'll be coming around. And I have your homework. I'll get it back into the class. We'll have some time to concept chat and then add a little bit more stuff today. So we'll do this another minute, and then we'll check in.
No, this is now on the, I spelled it wrong.
And what, Kyla, what is one thing that your group talked about that you thought was important so far today?
So we talked about the markers that we kind of went over, like the correlation and the neuropress cells. And then we started talking about how we swapped to kind of mammal-only things, like the embryonic stem cells.
Yep, yep. Juliet, what's another kind of thing you guys talked about?
We briefly brought up the colon, if that's how that's pronounced, which is the fluid-filled body cavity that
Yep.
contains our internal organs.
Yeah, it's phonetically it looks like that. It's pronounced coelom or salome, and coelomic cavity. Yep, yep. Okay, so we're going to keep talking about new stuff. This other thing we want to pick up from mammals. That's an experimental strategy. But do people have questions about anything at all?
Oh, I do. Question, chapter 6, where is it? Right here. Okay. In chapter 6, when they're talking about fertilization of the nematode, it said that polar bodies were formed after fertilization, and I was curious of like what that meant, because typically like that's all in like oogogenesis and like polar bodies and whatnot.
Yeah. We're going to talk some about fertilization and polar bodies in the frog next week because we're going to drop down to that molecular level. But just to mention, specifically this. So in intro-bio, you learned that, we have cells going through mitosis and dividing, making more cells. And then we have meiosis. And that's for cells that are going to make sperm and egg. And those cells go through two cell divisions, meiosis 1 and meiosis 2. And that's all. It was left there. It turns out that a mature egg in any animal species has gone through either all of meiosis 1 or 2 or part way, and it's stopped. And it finishes meiosis after fertilization before the sperm and egg nuclei fuse. And it's another example about how the animals in a given group all stop the egg meiosis process at the same point. So that's what they were talking about there in the nematodes, is that the nematodes stop at a distinctive spot and meiosis, and that's got to get completed.
Gotcha.
But it is a fertilized embryo. It's a one-celled fertilized embryo with both the sperm and egg in it. But in some cases, the egg nucleus has to divide again before the egg and sperm can fuse. So that's that. So, okay. Other questions people have? Yep.
Could you go over the 5 fingers analogy once more?
Yeah, absolutely. And we're going to get this during gastrulation, and gastrulation is referred to as the beginning of morphogenesis. We're going to start to fold the cells together to make a body. And so the mesoderm needs to be inside, and it often sort of starts out on the surface, and we sort of have to fold it in. And so we do that with the frog, with the blastopore, or we do it with the chicken, with the primitive streak. So things are folding in like that. And as they fold in, we can follow them inside. And so if we sort of thought of we could think of the blastopore as a hole like this, and here's some mesoderm. It's going to fold in like that. So it's folding in as because the cells are jostling, the valve's getting narrower, and they're getting this convergent extension. As it folds in, we can see it's a big chunk of mesoderm, but we can start to see that in that chunk there are these sort of 5 lines or chunks, strips, hot dogs, fingers of tissue that are going to play important different roles. And the middle one is going to be the notochord, and then next to it on either side are the somites, left and right, and then next to them are the lateral feet mesoderm. And so it's not a hand, it's not fingers, it's like hot dogs, it's not hot dogs, it's just these sort of slivers of tissue cells, and they're going to have different fates. They're all mesoderm, but those are really famous different mesoderm roles. And so we talk about each one in turn. Oh, notochord, right down the middle, right down the very midline of the organism. That's going to induce the central nervous system. That's the role of the notochord. Somites, they're going to get sort of sliced up, segmented up, like slicing a loaf of bread, and we're going to count them, 1, 2, 3, 4, 5, and the parasomites is going to give rise to each of the vertebrae in our vertebral column. We have this very anterior posterior organization of our body plan based on the numbers of the somites. Then the lateral pate mesoderm starts out as a hollow, but both of them are going to starts out as solid, but both of them are going to hollow out, and they're going to fuse together, and in this region they'll make that somic cavity. Other questions people have? We're doing great. Okay? So now the next thing, and again our book talks about it, and we want to get something out of it too, and it'll be great. The next is kind of a experiment where they say, oh, you just explained to me that's how a mammal develops, that pre-implantation, the cells are equivalent, they're embryonic stem cells. If we split the embryo, we could get 2 embryos, 2 identical twins. And so here's the experiment in test tube in a lab. Here they take 2 embryos from different parents, and they show them as being different by having a different color. And what we have is this mom and dad have white fur, and that mom and dad have brown fur. And they take these early pre-implantation embryos, 2 different, right there, really different mice, and they just squish them together to make one ball, and that will develop into 1 embryo. So in the same way you could take one embryo and split it to make identical twins, you could take 2 different embryos and join them to make 1, and people call that a chimera.
Is there any risk of like rejection of like one of the like?
No, because it's way before there's an immune system.
Gotcha. Okay.
Yep. Yeah, and it works just fine. And so it's a general term, it's called a chimera. And a chimera is an old word from like Greek mythology, where it's like that thing that's got a horse's head and or a human head and a horse's body, those kind of hybrid animals. And so it's not a monster, it's a perfectly normal, happy, fertile mouse. And they use that color pattern just because the fur color pattern is random. It's random in that animal's body which embryo gave rise to which. They could each give rise to each. And so here's just a litter of them where they created a bunch of those little chimera, and you can see each one has a different fur coat pattern. Just as a, for you to appreciate that. The cells don't merge together. The cells are separate. So there are some cells from the mouse brown embryo and some from the white embryo. And that's true throughout its body. Some of the cells of the liver might have been made by the mouse one, the brown one, some from the white one. But it's just in the fur that you can see this, but it's a good analogy. So it turns out that if you took those, say, 9 mice, and you bred them, each mouse, that mouse right there, each mouse makes either sperm from the white furred parents or the brown furred parents, the male, or eggs from either the brown furred or white furred. Because it just depends on which of the parent embryos gave rise to the cells that can make sperm and egg. So it's just this generation that is that chimera mix. And then their progeny are only gonna be from one of the original parent sets. There's no mixing together of the genomes and the DNA ever, not sharing of chromosomes ever. It's just this temporary time like that, all right? And so what's going on here is the scientists are like, great, that's just a simple experiment, and it's kind of surprising to see that's how it works. So what they realized is you could take two natural embryos, or you could take a natural embryo, from a mom, pre-implantation embryo, and then if you had a really special mouse cell, you could just add one cell. And we're going to talk about how they create these special cells. We could just add one special cell, and you could make a chimera. And if you made several independent chimeras, and they had babies, there would be some babies whose entire body was made from the special cell. So it's a way of converting a special cell that you have in a dish into a whole mouse that forever breeds true as that type of mouse. Okay, like that. And just an example, not yet of the experiment, but of natural biology. There are a lot of different types of cancers, bone cancer, skin cancer, and there's a very rare kind of cancer that's called an embryonic carcinoma, or teratoma, and it's in the testes or ovaries. It's not testicular cancer or ovarian cancer like you've heard of, because it's very rare. And they're very curious cancers. It's a solid tumor, so the root tumor could be removed surgically. And in that tumor, if you slice it, in those tumors, you find swirls of hair and skin and teeth. And that's very different than other types of cancer. In other types of cancer, when you slice the solid tumor, it just looks like a blob of cells. It doesn't look like brain cells or breast cells or skin cells, it's just a blob of cells. So these embryonic carcinomas are very strange. And just to give you an example of the curiosity of this little thing that we're taking advantage of, you can take a cancer cell, an embryonic carcinoma, that cell, you can join it like this to make a chimera. You can breed those independent chimeras and find another, an offspring that is just made of cancer cells, and that animal does not have cancer. So it's very interesting biology that scientists have learned a lot about. And that's one example, is that process is able to heal that cancerous cell and make it normal, and it can just be the whole body. And those guys, those offspring, and breed them, and they don't have problems with that kind of cancer or any other kind of cancer in particular. They're just regular mice. Yep. So we're going to stop there. We're going to talk about this a little bit, but we're going to be done with chapter 3. So next week we'll be rolling into chapter 4, and we're going to drop down to that molecular level. And that's important because 2 weeks from today we have our first exam. So next week we're going to be still learning a lot of new material. We'll have one more assignment to do like that. And I'll find my bag of stuff for this. But if you have art materials at home, you can